Alright folks, it's Pedantic Skeptic here, and we are going to keep rolling on with the Cornerstone Church's Apologetics session 35, and it's about uh, origins and evolution. I did see that they just uploaded part 3 of Origins and Evolution. So if you remember in my last video, I had seen that the part 2 was now like 9 days old or something like that, and they didn't have part 3 yet, but they said they were going to talk more in part 3. Part 3 is now up on YouTube, and the main reason I say that is because I don't want people to think that I am misrepresenting anything about the people that I respond to. I want to be kind of as honest and transparent as possible, and so my statement yesterday, while it was accurate, the on the day that I actually uploaded that video, they had put out part 3, and so even though, when the video was put out, it, technically part 3 had been put out. I mean, I don't think that people are going to watch, there aren't going to be a lot of people watching my videos the exact day I put them out, but just as a means of being completely transparent and honest, I was saying that, hey, now their video, part 3 on that is out. So yeah, that will be responded to at some point. I don't know how much of this shit I can take. So last time we got a little bit into how they figure out how different genes or different genomes are related to one another. And so what we're going to do is we're going to backtrack like 30 seconds in their video because I got distracted when I was trying to explain. Is somebody knocking on my door? I feel like I hear somebody knocking on my door. Never mind. It's fine. Okay. So, yeah, somebody's at the door. I'm not going to worry about it. I'm going to be continuing my fern transfers, and we'll be responding to Cornerstone. Alright, so let's rewind just 30 seconds. Okay.
After they broke it down into chunks to make a comparison, they excluded 25% of the human genome. So they just threw out 25%.
I don't know where he's actually getting that from. I don't know if he's actually reading the methods of the paper. Generally speaking, when they're making comparisons of how closely related two genomes are, they will oftentimes exclude things like DNA from like retroviruses, as well as things that might have been the result of contamination. So when you're taking samples and you're running the samples, there's a possibility that by you touching any of the equipment, you put some of your DNA in there, and so there may actually be too much human DNA in the human DNA sample, and so they factor for that by removing the human DNA. I don't know the full story because he's not actually showing us, and he doesn't actually, in the description of the video or anything like that. Actually, let's just double check the description of the video because I want to see. Let's see, I don't want to go to the chapters. What? Dramatic, intense trailer. I love how our church is using an artist called Heavenless, but that's okay. So nowhere here does he show us where he's getting this information from. Now, if I cite an article, generally, or if I'm citing the methods of an article, and this is what he's doing, like, if I cite an ecological principle where I talk about, like, oh, well, we know that, like, when, moth caterpillars smell a predator, or when they see a predator, they, eat less and they die at a higher rate. I mean, there are, like, 30 papers that address just that, and I may or may not leave a reference, but that's not as important as if I'm specifically saying, like, okay, there was a paper where they were looking at this, and their methods were blah, 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 and, maybe the reason I'm bringing that paper up is because I want to criticize the methods. If I'm doing that, I better have the fucking citation so that somebody can independently verify that I'm adequately explaining the methods. Now, I'm not a geneticist, so I don't know about how genetics work is done, and I'm sure this guy doesn't either. But he makes it seem like, oh well, we removed 25% of the human genome before we compared, so then it's not actually a 2% difference between the human and chimp genome. It's a fucking 27% difference or whatever. I think that's what he's implying, when that's not the case.
Of human DNA, and they threw out or excluded 18% of chimp DNA.
Okay. So I'm sure there are reasons why they did those exclusions. It probably has to do with again, like introns from retroviruses, or things that we know are non-coding, or maybe even things that are just not necessarily useful in making comparisons. The other possibility is they were excluding unique genome sequences between the two species, which sounds bad, although when you think about it, if there are things that are unique to each species, and then you have genes where, like those species has some form of that gene, if those species are far apart, the genes that the two species share, even though there's the same gene or their gene analogs where they kind of do the same thing, even though they have different sequences, should be further divergent from each other than species that are more closely related. So again, I don't know all of the nitty-gritty of this, but I suspect that excluding a little bit of the genome here and there for certain reasons isn't as nefarious as he's making it out to be.
These were things like repetitions and
Okay, okay, let's see what he says.
in the DNA and stuff that they just decided wasn't important.
It's not that they're not important, it's that they're not relevant. There's a difference between importance and relevance. And, I don't know if this study specifically was trying to get at, hey, how closely related are humans and chimps? I suspect that the bigger point was, what are some of the commonalities in specific regions of the DNA?
And then they took all the parts that didn't match up or make sense and just threw them out.
Is he going to expand upon this?
Called junk DNA. Yeah, they called it junk DNA.
Oh yeah, we got basically this far. So junk DNA is kind of a misleading term. It basically is non-coding DNA, which doesn't mean it's junk, but that's kind of just what people call it sometimes. It's generally the reason why they exclude non-coding DNA in some studies, not in all studies. There are some studies where they do want to know about non-coding DNA, but one of the reasons why they often exclude non-coding DNA is because they want to kind of correlate how the coding bits of DNA line up in species that have a lot of similarities. So what they might do is they might look at species that are pretty distantly related and keep the non-coding DNA to see just how far those species are separated. And the way that works is basically, if you imagine geologic time, the longer period of time two species have been apart from each other from a genetic perspective, the more divergent their non-coding DNA should be because of mutations. Meanwhile, species that are more recently diverged from each other are going to have the non-coding sections of their DNA be fairly similar still. And the reason they would look at the non-coding DNA is because the non-coding DNA doesn't generally do much in terms of, it's not really something that natural selection acts on all that much because it is non-coding, and the only things that non-coding DNA would actually affect are like viruses that attach to the DNA.
Which they're actually finding actually has function.
And yes, I've said that, I don't know if he's going to get into what the function of the non-coding DNA is. When it's called non-coding, that doesn't mean it's not useful. It just means that it doesn't actually cause a protein to be formed. DNA can do things aside from code for proteins, and so I think that's where the conflation here comes. And I don't know if he's doing it intentionally. If he is, shame on him. And if he's not, I really don't think he should be talking at length about this subject without proper knowledge of it. Um, I'm not going to necessarily talk about things that I don't have at least some familiarity with, and that's why I'm hesitant to talk about genetics, because I'm not a fucking geneticist. I study, ecology, plant physiology, and things along those lines. So if you want to talk to me about those concepts and principles, be my guest.
As science progresses, it actually hurts their arguments more and more.
What arguments are they making that science is hurting?
As we discover more about the human genome. But they excluded all that elements. So what would happen if you included all that, right? How similar would our DNA actually be? Well,
that depends, because if you include a whole bunch of things that are there, you might end up, I don't know, including things that are the result of contamination or a bias in the way you extract the DNA.
If you don't exclude the dissimilar chunks from the comparison, it's only between 66 and 86% similar.
Okay. Again, that's still quite a bit of similarity. I don't know exactly where he's going with this.
It's a big difference from 98%. I mean, when you think about what DNA is actually controlling, it's controlling cell function. It's
so that's actually an interesting thing. So let's just assume that humans and chimps share 98% of their coding DNA, which is really fucking huge, and their total DNA, which includes the non-coding bits, is only 67% similar. That is still quite a bit of similarity, especially given, the human lineage and chimp lineage split from each other, I think at least 3.5 to 4 million years ago. It's scary that many differences, mutations can accumulate, especially when you consider that, I think chimps take 10 years. Actually, let me look it up so that people are, let's see, age of maturity chimpanzee. Okay, so chimpanzees start making babies between 3 and 14, between 7 and 14. Well, yeah, they make their babies at about 14 years old. I'm not saying humans should make babies at 14, but hypothetically they can. So, if you think that a generation takes, let's just pretend, actually, when did the chimp and human... Oh boy. Okay, so our ancestors have been apart for about 8 million years. Let's just say that it's 8 million. Where is my fucking calculator? So 8 million years divided by, Jesus. And divided by, we'll just say it's about 15 years per generation. That's still like 500,000 generations of separation between us and chimpanzees. And, it's a little scary that many mutations have occurred between humans and chimpanzees for there to be that much of a difference in the non-coding DNA in that few generations. Relatively few, I should say. 500,000 isn't a few, but compared to things like insects where they have, a 3-month generation time. Not every insect has a 3-month generation time. I'm just kind of using one example, but there are plenty of insects that have longer, plenty that have shorter generation times. Anyway, that still is a good number of mutations. And one of the things they do is called molecular clock, where they take two species that are different from each other, and they figure out how many differences there are, and they determine, like what on average is the mutation rate for this DNA. And based on that, they can figure out how many generations it would take for that number of mutations that they see to actually accumulate, and then by doing that, they can figure out how many years ago the two species diverged. And of course, it's not perfect. You get a range because you don't know. You're using an average mutation time. You're using an average generation time. You're also assuming that the mutations are accumulating at an even, consistent pace. And so it's not supposed to be perfect. It is kind of just a rough estimate. It's cool though.
Actually, it's actually code that tells our cells how to function, right, how to work.
Other things that they do is they provide like areas for viral DNA to attach to, and they provide regions where RNA knows how to attach, and they also provide spaces for like the cellular machinery that mediates cell replication and things like that to attach. It's not that junk DNA is useless. Junk DNA is non-coding, which means it doesn't get directly translated into a protein. That's very different from something being useless.
So it's actually a really big difference when you don't essentially cherry pick the parts of DNA that you want.
Nobody's cherry picking. The claim is that the code, they say that, we're, 98% genetically the same as chimps. I mean, that's kind of layman's terms, and in science we don't use layman's terms for the most part. If I were to find an article where it talks about coding versus non-coding DNA, they would probably make that distinction. And it seems like I'm nitpicking here, but there really is a big distinction when you want to talk about, coding versus non-coding DNA. And so, these people are, I don't know if I don't want to accuse him of deliberately misconstruing what's actually happening. However, I will say it does seem a little bit like he is, trying to misrepresent what the science is actually saying, because science isn't saying necessarily that, the entirety of the human genome is only 98% is 98% of the chimp genome. Even though people like to say, oh well, we share 98% of our DNA with chimps. That's not entirely true. They generally, when they say that, mean 98% of coding DNA, which again is different. But that's not a problem with science. That's a problem with people talking about science and being inaccurate about it. Okay? If anybody's ever read a pop science article and then read the actual science article that pop science article is written about, you know exactly what the fuck I'm talking about.
To actually compare. So it's a big difference. And humans supposedly branched off from chimps about 6 million years ago.
7 to 8, actually. Now, here's the thing. If this video was made a few years back before we, kind of made some of those discoveries that led us to the number, to the 7 or 8 million years, I'd be like, okay, it's a little bit of outdated information because, he was working with the information we had. But this video was put out about 2 weeks ago. Okay? I don't think the Google search of, our genetic lineage split is actually just a couple days old. I think that he is and like the exact number doesn't even matter, but just let's continue on.
And that 6 million years isn't enough time to account for the random mutations necessary.
Actually, we should let's check this. I don't know if he is okay, one sec. So humans have let's just see something here. God damn it, where's my calculator? Just a sec, folks. Let's calculate this right now. So 3.2 3.2 billion. 3.2 billion. Okay. So if 67% is the same, that means that 23%, no, 33% is different, times 0.33. So that is 1 billion 56 million mutations. Okay. Average mutation rate for humans. Okay. Just trying to see. 175 mutations per diploid genome. So let's just think 175 mutations per genome, right? So let's just divide this by 100 and, what was it, 175? We get at 6,034,286, roughly. Generations are required to have that many mutations. However, what you have to remember is that mutations can oftentimes follow each other. I mean, there are a couple of things going on here. The numbers don't add up, which I'm not surprised at, but you have to think. I don't know how many chimp and human genomes they looked at to come up with these. The other thing to remember is that this is the mutation rate that's occurring from one individual. You have random mating of multiple individuals through each generation. So we could effectively divide this even further because of the fact that, this is from one pair. It's hard to explain, but basically what we're looking at here is you have, you need about 6 million generations if the starting population of these organisms is, one pair of mating individuals. And that's not exactly how that is working. The mutation rate is kind of tricky because you are you have to apply it to multiple individuals. Again, I'm not a genetic scientist, so I don't know exactly how all this works. It's really complicated. It has to do with, what mutation rate you expect to see over each generation, and you have to then extrapolate that to the entire population, not just every individual. Because if you have an individual with, several mutations, meaning with an individual that has several more mutations, you have to add those number of mutations up in order to get how divergent that offspring is going to be, and then you have to confound that with every additional offspring they have and every additional mating that occurs during the generation, and it just becomes a giant mess. And it's not something that I have really any practical or technical experience in, and so I it's not that I can't fact check him, it's that actually, let's just see. Claim. Oh, intergenerational mutation rate does not equal long-term evolutionary substitution rate. Oh, crap. Didn't mean to do that.
Human genome. So they just threw out 25%.
Fucking shit.
Don't exclude the dissimilar chunks. From the comparison, it's only between.
So anyway, he's using the mutation rate to measure something that you actually have to use a different calculation for. It's like trying to measure the distance from, the Earth to the Sun using a scale.
66 and 86% similar. It's a big difference from 98%. I mean, when you think about what DNA is actually controlling, it's controlling cell function. It's actually code that tells our cells how to function, right, how to work. So it's actually a really big difference when you don't essentially cherry pick the parts of DNA that you want to actually compare. So it's a big difference. And humans supposedly branched off from chimps about 6 million years ago. That 6 million years isn't enough time to account for the random mutations necessary for that much difference. This is why it's so important for them to get to 98%.
Again, I just said that the mutation rates, not the way you actually measure, how long ago things. I was talking about molecular clock. I think there's. I don't know if you're actually using mutation rate or something else, but the molecular clock stuff is a complicated topic that I don't have much authority to speak on. Neither does this guy, by the way. But I think that if we were to contact somebody who's actually experienced in how this sort of thing works, that person would be able to say like why what this person is saying is not true.
Because if you've only got 6 million years of random mutation, and they've got formulas for how much time it takes to create a new species from.
So we'll just assume that like there's a set definition for species, and speciation can occur when enough genetic differences exist between two populations. Let's just assume that that's decently accurate, not 100% accurate, but close enough. One of the things you have to realize is that when differences add up in coding versus non-coding portions of that genetic code, you're going to have differences in how quickly two populations diverge and become different species. If differences in the DNA occur on portions of the genome that control like the shape of the genitals and how the sperm is coded and things like that, you're going to have much more rapid speciation because a reproductive barrier is going to form a lot more quickly than if the mutations are occurring mostly in non-coding spots. So different groups are going to speciate at different rates, and it's kind of a case-by-case basis. And the problem with that is because there are so many species, when you calculate average mutation rate across species and use that to extrapolate how many generations you expect a population to diverge into different species, without even considering the starting population size and shit like that, you are really not going to get a very precise or accurate measure of how quickly these things are diverging from each other. It's a very complicated science, and there's a lot we still don't know about it.
From a previously simpler form, and 6 million years, not enough time for only 66% or 86% difference. That's why they have to get to this like 99, 98% number.
Did he just say there was a 40 to 60% difference? It's a 40 to 60% similarity.
Man, the whole problem, we talked about Occam's razor. Right?
Yes, Occam's razor, yep.
Because you just said, well, there's an assumption about time, which is improvement.
Right.
There's an assumption about this is how long it takes for mutations to create new species, which is another assumption on top of an assumption.
Based on.
Again, it's a calculation. And like I just said, it's not the best, and it doesn't prove creationism at all. It's just that we go ahead and we, what the fuck is this? Oh, that's, we go ahead and we, make these, calculations that are based on averages that could be way off because we have such a spread. So, here's an example. I just gotta keep an eye on the time. Let me open a new Excel sheet. Come on, give me a new Excel sheet. God damn it. There we go. Okay. Actually, no. Let's, okay. Actually, fine. Okay. So, let's say let's say we look at, a couple of species, and, this is the mutation rate for each species. That's an average of 10. But let's say we look at a group of species where these are the mutation rates. The average is still 10 if you look at the, on Excel, if you look at the bottom of the bar that, has this, it will tell you have an average of 10, a sum of 9, count, or a sum of 90, count 10, or 9. And here, when you do the same thing, sum of 90 again, but the average is the same, but you're all over the fucking place. The spread of your data is different in these two data sets, even though the average is the same. So you can have averages that really, really diverge from, the highest and lowest points. And so using average mutation rate isn't the best way of calculating, how quickly things could have diverged from each other, especially in certain groups where, maybe the mutation rate is super high because of, a very short generation time or something like that.
On an assumption, yes.
Based on assumption, and then you get to what you're talking about. But that's only part because you also said, well, where does life come to begin with?
We're going to get to that. We are going to talk about chemical
So
evolution.
The layers of assumptions and theories
Sounds kind of like faith, right?
are incredible. Well, absolutely incredible.
That just brings up the whole fallacy of peer review.
Yes.
Because peer reviews cause this problem.
Yes, yes.
Peer review is actually a process by which we eliminate problems because we point out biases that people who are writing any kind of paper, oh crap, I inhaled some of my own spit, may have had. Okay.
For sure.
Yeah.
Because you go in with a presupposition,
Yeah.
right, that we evolved from chimps. Like,
No, we, oh fuck, why is this? Okay. Come on.
we take that.
No, the presupposition, not that it's a presupposition, but the idea is that we have a common ancestor with chimps and our lineage is split apart. We didn't directly descend from chimps as far as we can tell. And, regardless, the use of the word presupposition here implies that there's absolutely no evidence for this. And I feel like he could use any, species relations to make the same claim, which makes his point kind of weak.
That is fact, and then you have to kind of work the math or the science or whatever it is to try and make that truism true, right? And so that 6 million years is a problem.
again, it's about 8 million years, but regardless, I don't know exactly how these calculations are done. I'm sure if I wanted to, I could, go through the literature and read the original articles that are, suggesting these numbers. Remember, the mutation rate isn't necessarily the best way of determining how long ago things diverged from each other. The what's it called? I forget what it was that I said it was. But there are other factors, and the reason for that is because mutation rates assume that single individuals are giving rise to later individuals in the population, which isn't how that works. Um, and you might be saying, well, if there's this much difference within the whole chimp population, shouldn't it take longer for that difference to proliferate through the population? Not necessarily, because you have random mating and reshuffling of genes, as well as mutations that occur in the gametes that don't occur in the actual somatic cells. And you also have the issue of mutations not necessarily being independent of each other, and that's not something I'm going to delve into because I'm not a geneticist, but there are times where one mutation can make other mutations more likely, especially if it's a mutation in a DNA repair protein or something like that, where basically when there's a mutation, most of the times it's caught by different functions as the DNA replicates. But if there's a mutation affecting that, then other mutations can come along more rapidly. The other assumption that they're making is that the mutations are only occurring in the non-coding portion of the genome, and that's not the way that this works. But in any case, the problems that we run into with their representation of this is this guy doesn't have a good grasp in it. I don't have the best grasp on it either, and I'm probably better versed at biology in general than this guy. Again, it's not my specialty. You want to talk about ecology, plant physiology, insects and plant interactions and things like that, then we can have a conversation. But when it comes to, like genetics, mutation rates, molecular clock stuff, I'm just, fairly clueless. And so is this guy.
Actually, the Big Bang, just to lean back on the origin of the universe, the Big Bang was a huge problem for evolutionists, right?
No, it actually wasn't, because evolution doesn't deal with the Big Bang.
Because in Darwin's day, they thought the universe was eternal. Plenty of time.
Again, eternal and infinite are different things. The universe, if it began when time began, is technically eternal, okay?
Plenty of time for random mutation by natural selection. But now you've posited, even if you go with the Big Bang, age of 13.4 billion years, and Earth's age of 4.6 billion years, right? Even if you go with those numbers, well, now that gives you a much different time.
So first of all, this guy hasn't even mentioned the fact that, it's only been about 1 billion years since multicellular eukaryotic life has been on the planet. The first, I think 1.5 billion years of Earth's history, there was no life. So the fact that he hasn't brought this up, and I'm not saying this as a point for him, what I'm saying this as is he doesn't have a good grasp on what the science actually says. But let's just assume that the moment the Earth formed, the first life form arose. 4.6 billion years, especially when for the majority of the history of life on Earth it has been unicellular organisms that can multiply several times within an hour, that is plenty of time for mutations to build up to give rise to the diversity that we see. Okay? I could see, because I'm sure he believes that the Earth is 6,000 years old. If that's the case, then yeah, that's not a lot of time for most things to diversify, unless we're thinking about, insects and microbes. But, at the same time, the thing he's not recognizing is that mutation is not the only way in which new genetic material arises, and natural selection is not the only way at which populations evolve.
Time frame to deal with than eternity, right? Because you're dealing with rolls of the dice, right? These mutations are random. They're not.
More or less so. I wouldn't say they're completely random, so to speak. What they are is usually a replacement of it's an error in the DNA replication process, and sometimes beneficial mutations or harmless mutations can accumulate because they keep getting passed down, and so it's not
guided.
Well, it's not like every time a mutation occurs is an independent event.
By any intelligence. The only intelligence in Darwinian evolution is the survival of species, right?
It actually relies on the extinction of things, but that's just a nitpick.
Darwinian evolution propagates the random mutations that work. If the organism dies,
No, it propagates the mutations that don't kill you off. Which I guess, yeah, it stuff that works, but it's not necessarily only the strongest, whatever percentage, the top 10 or whatever survive. Look at the human population and how many dumbasses have kids these days.
There's no accumulator that's saying, oh, that was a bad one, let's not do that one again, right? The roll of the dice is from scratch again.
Not always. In fact, there are a lot of organisms that have various genes that account for or that take care of mutations that occur. And you might be thinking, well, then doesn't that slow the mutation rate? That depends. It depends mostly on where that mutation has occurred and what the exact factor for suppressing mutations is. Some of them don't work at the DNA level. Some of them work at the actual protein synthesis level, where they get rid of proteins that aren't supposed to be there after the fact. Which is sort of an energetically wasteful process, but if that organism is still able to pass its genes on, then those genes get passed on.
So the organisms have to survive to propagate the good mutations. So the thought is, bad mutations, organisms die off. Good mutations, organisms live.
That's a false dichotomy, because there can be plenty of neutral mutations, and good and bad are relative terms. So, mutation that, makes a plant develop a waxy gloss over its leaves, and that waxy gloss reflects off extra sunlight. That's good in a fucking desert. That's probably bad at the forest floor, because the plant's not going to get enough sunlight. And we haven't even touched something called phenotypic plasticity. So let me illustrate phenotypic plasticity really quickly. Okay? No? Okay. I like to use this as an example of phenotypic plasticity. Okay? So, this is the pink lady slipper. I know the flower looks like a scrotum. I call them scrotum of the woods. I have a bunch of them growing. But this Acale, Cypripedium Acale, is in bloom. It's at the same stage of its yearly cycle as these guys. But look at the difference. These guys are in an open, exposed area where there's a shit ton of sunlight hitting them. You can tell just by the picture. This picture of Cypripedium Acale, they're not getting nearly as much sunlight. Look at the difference in those leaves. Not only are the leaves and the plants that are grown under the sun much, much narrower, they're angled at a different way, and they're much lighter in color to reflect off more light. These guys have these broadly dark, and they're catching as much light as they possibly can. And that's phenotypic plasticity. That is, the same organism is capable of displaying different traits. Another example would be some of the hydrangeas with different color, flowers, depending on the pH of the soil they're growing in. But by the way, not all what's this? Okay. Anyways, let's not worry about that. Where were we? So, phenotypic plasticity is the same organism able to go down different pathways depending on what the environment it has encountered is. And because of phenotypic plasticity, there are mutations that don't necessarily do anything good or bad. It's more situational.
the survival of the fittest, right? Good mutations, organisms live and propagate those mutations to the next generation, and then on and on and on it goes, right?
Not exactly. We can talk about the different methods at which the different mechanisms in which populations can evolve that don't involve natural selection. You have things like genetic drift. You have things like what's it called? Artificial selection. You have the founder effect, which is a type of genetic drift, and bottlenecks are another type of genetic drift. Things like that, wherein populations are subject to various things that aren't actually natural selection, that cause differences in their genomes as they age, as the population ages and continues to grow, and that's evolution. It's just not through natural selection.
And so, if you have a limited amount of time for this roll of dice, right? There's probability on if I'm just rolling the dice randomly.
Okay, but you have to consider that in probability, the law of addition is, hey, you have all these genes in the genome. Let's just play around with the calculator for just a sec. Or let's draw this out real quick. Where the fuck is Microsoft Paint? Jesus Christ. Paint. No, not that. Paint. So let's say, no, I don't want to use that. Undo. Let's say this is a DNA strand. Okay. We'll use circles to represent A. Close enough. God damn it, I want this to be touching the backbone. Circles are A. Diamonds are going to be T. This doesn't have to be perfect. And then we have C and G, which are going to be these two shapes. Right? Doesn't really matter. It's so this is a strand of DNA. So let's just say there is a 1 in 4 probability that any given nucleotide is going to be different. It's going to be a mutant. Okay? So you have 1/4, 1/4, and I'm not saying that this is the actual probability. DNA errors actually occur a lot less frequently, but let's just say that this is the error rate. But then you got to do it for the other side. And also, because, it's the probability that the mutation will happen here, or here, or here, or here, or here, here, you add all those probabilities up. So let's actually go, I think it's DNA polymerase error rate. Okay. So once every 10 to the 4th. So that's going to be a 1 in 10, I believe, with 4 zeros. Okay. However, each time a cell replicates, there are between 100,000 and 1 million errors that occur. Okay? Now, usually those errors will be caught and corrected. But let's just pretend that you know, 0.00001% of the time, they're not. Okay? Oops, I don't want that anymore. Don't save. So, we have a 0.001% chance that there is a error. We're going to multiply that by itself, 0.0001. Okay. Now, remember that there are going to be several million. So the odds of a mutation going uncaught, unfixed in, let's just say a 1 million base pair strand of DNA. That's still 0.1% chance. Now remember, there are going to be, let's just say there are 1 billion cells in that organism. And now you have 1,000 mutations that occur throughout that organism's cellular history, cellular replication cycles. Although it's probably going to be a little bit lower because, every time you replicate it, I think there are like 40-something cell replication events during an organism's life, depending on the organism. But then you got to multiply that by all the organisms in that population, multiply that by the number of generations of that organism, and as you'll see, you'll get plenty of mutations occurring. It's, mutations are relatively rare, but because of how many chances there are for mutations to occur, they're common.
And nothing's guiding that process. I need a large amount of time for the random mutations to basically hit the lottery, right? And for this evolution to actually occur.
This sounds a lot like an argument from incredulity.
And so, this next video I'm going to show you is actually a video not from a Christian source that talks about the 98% and tries to explain why, this stuff was thrown out and it's really okay that it was thrown out and all that stuff. So I thought it was cool to show it. So let's show it. Let's see. Can I just play it?
I would like to see
It's often said that we humans share 50% of our DNA with bananas.
Let me just see something here. Minute physics, no. God damn it. Minute science, chimp DNA. Minute Earth, let's see.
Research shows nostalgia can help you remember ads. So customize and save with Liberty Mutual.
Jesus
The thing that's different about a Verbo vacation home, you always have the whole...
It's often said that we humans share 50% of...
Actually, we're gonna see that anyway. Well, that's gonna be quite a while back. But anyway.
80% with dogs and 99% with chimpanzees. Taken literally, those numbers make it sound like we could pluck one cell from a chimp and one from a human, pull out the tangled bundles of DNA known as chromosomes, unroll each one.
They're not just tangled bundles of DNA, there's other stuff in a chromosome like histones, but that's not really important to know.
Like a scroll and read off two nearly identical strings of letters. But in reality, the human and chimp scrolls don't sync up so easily.
Yes, this is correct.
In the 6 to 8 million years since we split from our last common ancestor, chance mutations and natural selection have changed each of our genomes in radical and unique ways. Two human scrolls fused, leaving us with 23 pairs of chromosomes to chimps' 24.
Yes.
Other large mutations revised huge sections of text, duplicating a chunk of human DNA here, erasing a chunk of chimp DNA there, while throughout the scrolls, tiny mutations swapped one letter for another. When researchers sat down to compare the chimp and human genomes, those single letter differences were easy to tally, but the big mismatch sections weren't. For example, if a genetic paragraph, thousands of letters long, appears twice in a human scroll, but only once in its chimp counterpart, should that second human copy count as thousands of changes,
Yes.
or just...
That is a good question. What I would say is, you would count how many, so basically that's a gene duplication, and when gene duplications occur, there are opportunities for mutations to occur, and so this is actually how eyes can have different color cones, because instead of having one eye protein pigment gene, you now have, two, and one can be modified so that it reflects the color red rather than color blue or whatever. And, that's why we have the trichromatic vision when dogs only have dichromatic vision. Dogs are not colorblind, they just don't see as many colors as we do, but that's irrelevant. And so what I would say is the each divergence between those two should be counting, but not, every single, copied over piece.
Yes, they should.
And what about identical pairs?
Just also keep in mind that a few thousand letters out of a 3.2 billion base pair DNA still is like 1%.
Graphs that appear in both genomes, but in different places, or in reverse order.
Yep, that's an inversion.
Were broken up into pieces. Rather than monkey around with these difficult questions, the researchers simply excluded all the large mismatch sections, a whopping 1.3 billion letters in
That is a big chunk, yes.
all, and performed a letter-by-letter comparison on the remaining 2.4 billion, which turned out to be 98.77% identical. So, yes, we share 99% of our DNA with chimps, if we ignore 18% of their genome and 25% of ours. And there's another problem. Just as a small tweak in a sentence can alter its meaning entirely, or not at all, a few mutations in DNA sometimes produce big changes in a creature's looks or behavior, whereas other times, lots of mutations make very little difference.
Yes, this is another point that these people are not really getting into.
So just counting up the number of genetic changes doesn't really tell us that much about how similar or different two creatures are. But that doesn't mean we can't learn anything by comparing their genomes. DNA contains a record of the evolutionary relationships between all organisms.
Yes.
It's a garbled record, but by reading closely, we've been able to glean enough information to refine the evolutionary trees we started drawing long before genome sequencing was around. We may not actually be 99% chimp, but we are 100% great ape, and at least a little bananas.
That's funny. So you can see the presupposition in this video, even though, they were laying it out. They're not hiding the ball here, right?
So one of the things that I do need to bring up is that this is probably an instance of pop science taking a scientific article, misconstruing what the actual scientific article said, and then lay people just rolling with it, and that's how that misconception just keeps going and going and going. That's how misinformation happens. It's not disinformation because it's not intentional. It is people who aren't scientists trying to communicate science to non-scientists, because there aren't a lot of people that can effectively do science and communicate it at the same time. But anyway, it's not necessarily scientists trying to be deceptive. It is scientists finding one thing and playing the telephone game with science communicators who then relay a different message, and now everybody thinks, something that's not scientific.
They're not hiding the ball. They're laying it out that they excluded all this stuff and that they couldn't make sense of this stuff, so they just tossed it. Nevertheless, we're certainly evolved from chimpanzees or great apes.
it's not that we're evolved from great apes. We still are great apes. The law of monophyly. And it honestly wouldn't matter how big or small of a percentage of DNA we shared with something, a common ancestor or something that we shared a common ancestor with. That's just a reflection of how the different evolutionary paths went after the split.
This video shocked me because it wasn't a Christian video. It wasn't like Answers in Genesis or somebody trying to argue for theism. It was essentially someone who accepted the belief in evolution as fact, laid out all the problems with the genome, which frankly the discovery of the genome and how cells work is really having a seismic shift in in evolutionary circles.
I hope he explains that. Expand upon that, please.
They have something called neo-Darwinian folks that have had a conference. Stephen Meyer talks about this in some of his videos, which we'll look at some of his stuff later. But talks about how they are basically saying we need to come up with a different theory of evolution.
A different theory, or do they need to just refine the theory? All right. So that was a whole lot of BS. We are 40 minutes into their 42 minutes into their hour and 6-minute video. So we're about 2/3 of the way done. Oh man, I'm tired. Jesus. They haven't made too many good points, but we're gonna just keep seeing where this leads. So thanks for tuning in.